Hi guys, and welcome to our lectures on the thyroid gland. In our previous lesson, we saw the overview of the thyroid gland. A quick recap, please. We said that the thyroid gland is one of the organs of one of the glands in the endocrine system. It is located in the neck, below the larynx, and in front of the trachea. Right? It is made up of it is a bilobe structure, meaning it has two lobes that are connected with a structure known as the isthmus, and within it is it's bilobe. We say it is also butterfly shape. It weighs about 25 grams, right, in weight, and it is made up of two types of cells. One of the cells is follicular cells that actually produces T3 and T4. Then we have the second group of cells known as parafollicular cells or C cells, and this group of cells control they secrete calcitonin. Now, basically, we'll be looking at synthesis, storage, and release of thyroid hormones in this lesson. But before then, we'll look at the stimulation, what actually causes the stimulation. So what I'll do now is just to have let's have, okay, so let's assume this is the thyroid gland, right? This is the isthmus, and then within this structure we have some cells, right? We have some cells known as follicular cells, all right? Follicular cell. Now, then, around this cell, or yes, around it, we also have some other cells known as parafollicular cells. Parafollicular cells. Now, so what we'll do now is that we'll zoom into this. We'll zoom into these cells and see how it looks like. That is a follicular cells, right? Remember, we say it synthesizes what? It synthesizes T3 and then T4. Now, these cells are made up of cuboidal epithelial cell. They are made up of what? Cuboidal epithelial cells. Okay, so this aspect is the lumen, right? This is the lumen, all right? And within this lumen, we have some structures known as colloid, right? Fluid medium known as colloid. We'll look at that in detail later on. So these cells, as I said, are made up of cuboidal epithelial cells. Now, this is just a typical example of a follicular cell. Now, actually, what actually causes the stimulation? What causes this cell to produce T3 and T4? Now, if you remember from the hypothalamus, the hypothalamus actually releases some hormones known as thyroid-releasing hormones, and these hormones act on some cells in the anterior lobe of the pituitary gland. All right. So let's assume this is your hypothalamus. All right, this is your thyroid gland. All right. Now, this hypothalamus releases some hormone known as thyroid-releasing hormone, and then there are some cells here. Those cells are called thyrotrophs, right? Thyrotrophs, and this hormone will act on this thyrotroph to release another set of hormone known as thyroid stimulating hormone. Okay, what are we seeing now? The hypothalamus secretes thyroid releasing hormone, which acts on a cell in the anterior pituitary gland to produce TSH. Now, TSH will act on what? Will act on the follicular cell, causing it to what? To start to it causes stimulation. It stimulates the follicular cells to begin to produce what? T3 and T4. So that's one of the basic stimulating factor. Then the second stimulating factor is known as the autoregulation. I will explain that later on. Autoregulation. Now that is, the follicular cell itself has the ability to regulate thyroid hormone production. Okay, so I will talk about that in a subsequent shortly. Now let's look at these cells, and let's look at it in depth. This I'll just take one of these cells, and then we'll look at it. Let's make it bigger. So we'll make this cell very, very big. So let's assume this is one of the cell. All right, this is one of the cell. Remember, we have a central portion known as the lumen, and then you have other cells like this. Okay, let me make it neat so that it's not confusing. All right, so let's assume we have something like this. All right, so this is the lumen. Now this is just one of the cells. We have a nucleus here. Now this area of the cell is known as the basal membrane, right? So this is what? The basal membrane. Then this side is the apical membrane, or the luminal side, right? You can call it the luminal membrane. Then this is the lateral side. Now, so if we want to synthesize T3 and T4, first of all, we need some raw materials. We need some materials, and one of the important raw materials that we need actually comes from the blood. And that is what? Iodide. Right? Iodide. Now remember that our food, most of the food we eat is rich in iodine, right? So when you eat particularly your iodinated salt, iodinated oil and all of that, now in the GIT, right, this iodine is converted to iodide. What did I say? That in our food, most of our food are iodinated, they are rich in iodine. So when you eat, right, they are absorbed in your GIT in form of iodide. Now about 20% of this iodide are taken up by the follicular cells. Then about 80% thereabout are excreted out. So that means, first of all, one of the raw materials that we need is what? Is iodine. Now at the basal membranes, or at this basal membrane, we have a pump, right? We have a pump known as iodide pump. We have what? Iodide pump. Now this iodide pump transports iodine, right, from the bloodstream. This is the interstitial space. Okay, so it helps to move what? Iodine across the membrane, because this iodine usually they are charged compound, they are polar compounds, so they cannot just cross membrane. They need like a carrier. So this iodine pump helps to move this iodide, right, from this surface, from the blood capillaries into the luminal side. Now, but there are some things I need to say about this iodide pump. This iodine pump has the ability to regulate. That's why we say one of the functions of the follicular cell is autoregulation. Do you understand? It's autoregulation. So what's happening is that if, for example, you have too much iodine, you have what? You have too much iodine coming in, this pump will be inhibited. Right? I know it's sounding somehow, right? Now, if I say it again, if you have too much iodine, right, coming from the bloodstream, it tries to inhibit this pump, try to reduce, or the activity of this pump is reduced, right? Now, when you now have reduced production of what? Of T3 and T4, right? Then the activity of this iodine pump is increased. So that is why we say the follicular cell or the thyroid tissues have two stimulatory mechanisms. Number one is the TSH, it's coming from the anterior lobe of the pituitary gland that stimulates the follicular cell to produce more hormone. And then the second factor is the autoregulatory function that is carried out by what? By the iodine pump. Now there are some also, there's a drug called perchlorate that also inhibits, right, perchlorate, right? Now this compound or this drug also inhibits the activity of this pump. Okay, so that means for people that are having hyperthyroidism, they can benefit from this drug. Now let's look at it now. So our iodine moves from where? From our bloodstream, right? And then it moves towards the what? Towards the apical side. So that means at this region now we have our iodide. Are we okay? So that means this pump, this iodine pump, just helps to shift iodide from the capillaries, from the capillary side into the luminal side. Then the next thing that we want to look at, right, is the fact that we have some polypeptides that are produced, and this polypeptide is called thyroglobulin. Thyroglobulin. Right. Now, since they are polypeptide or they are protein in nature, it means they are made up of a lot of series of amino acid. All right, they are made up of what? Amino acid. But the most important one that concerns us is tyrosine. All right. So let's assume that I have this as our thyroglobulin. Let's assume this is our thyroglobulin chain. If it's actually a protein chain, it will have lots of amino acid. So let's assume this one that is having this box like these are tyrosine, right? These are tyrosine. Then let's assume these are other forms of amino acid. These are other amino acid. Okay. Now, so the whole of this structure is known as thyroglobulin, but within this thyroglobulin chain, we have what? Tyrosine. Okay? Now, so all of these structures are also transported into the lumen. Are we together? Now, so that means we have iodine. We have iodine in the lumen, and we also have thyroglobulin in the lumen. So let's assume this is our thyroglobulin. This is our thyroglobulin, and then this is tyrosine, and let's say tyrosine. Okay. Remember, you have other forms of amino acid which are not important for us at this level. Okay, so what have we learned so far? That in the synthesis of thyroid hormones, some things are very important. One of it is iodide, and this iodide is moved from the blood capillaries via the iodide pump into the luminal side. Okay, now then we also have thyroglobulin that is synthesized within the cytoplasm of this follicular cell. Okay, and then this thyroglobulin is proteinous in nature, and that means it is made up of a lot of amino acid sequence or series. But the one that concerns us is these ones that are called tyrosine. Now, within this lumen, we have a special enzyme that, please, I won't advise you to forget this enzyme. The name of this enzyme is thyroid peroxidase, right? Thyroid peroxidase. What's the name of this enzyme? Thyroid peroxidase. Now, this enzyme is located at the luminal aspect of the cell. Now, first of all, this thyroid peroxidase helps to convert iodide to iodine. Do you understand? It helps to convert what? Iodide to iodine. And that process is known as oxidation. Alright? Let me do something like this. Let's assume I have my iodide here. Right? And then this enzyme, which is thyroid peroxidase, right, will convert iodide to what? To iodine. Do you understand? So that means the first process, the first thing that we need here now is that this enzyme has helped us to convert iodide to what? Iodine. Now this process is known as what? Oxidation. Oxidation is loss of electron. So our iodide has lost electron, but let us not go into all of those details. Now, so that is the first step. Now the second step, this same enzyme, thyroid peroxidase, now does what we call iodination. Iodination is also known as organification. Okay? So what's happened now is that we will attach, let me use a different color. Let's assume this is my iodine. I will attach one iodine to tyrosine. Okay? So if I attach one iodine to tyrosine, this compound now is called monoiodothyronine. Right? What are we saying? That initially we converted thyroid peroxidase, converted iodide to iodine. Then this same enzyme, right, thyroid peroxidase, carries out the next step in the production of this hormone, which is organification or iodination. And then we attach iodine to our tyrosine. So let's assume we want to attach 2 iodine, right? We want to attach 2 iodine to this compound. Then this compound now will be what? Diiodothyronine. Okay? Diiodothyronine. So let's assume we have another one again here. Monoiodothyronine. This is what? Diiodothyronine. Okay? Let's assume this is what? This is also diiodothyronine. Let me make it longer. Alright? We have so let's assume this is diiodothyronine and this is 1 iodine. So what are we seeing? That in iodination we attach iodine to our tyrosine. So if we attach 1 iodine to 1 tyrosine, it becomes monoiodothyronine. And if you attach 2 iodine, right, to a tyrosine, it becomes diiodothyronine. Now, so this is also done, or this activity is catalyzed by this enzyme, thyroid peroxidase. Now, the next step in the formation of these thyroid hormones is known as coupling. It's known as what? Coupling. Now, let's look at this one now. This is diiodothyronine. This is what? Monoiodothyronine. Then this same enzyme, which is what? Thyroid peroxidase, will couple these 2 guys together. Right? Will couple them together. Now, if you have 1 and 2, it becomes tri. Okay? So this compound is now called T3, which is what? Triiodothyronine. Then if you have like this one now, that is 2 iodine and 2 iodine, if these 2 guys are coming together to be coupled together, this same enzyme, thyroid peroxidase, couple these 2 guys, which is, this one is diiodothyronine, this one is monoiodothyronine. So the coupling reaction that takes place between the 2 guys, right, form a new structure known as triiodothyronine. Then in this case, this same enzyme, thyroid peroxidase, couple these 2 guys. This is what? This is diiodothyronine. This is also what? Diiodothyronine. So these two compound now becomes tetraiodothyronine, or you can also call it thyroxine, which is your T4. Now, so that is what goes on within this area, right? So there is oxidation, there is organification, and there is coupling, and then there is formation of T3 and T4 in the process. There is oxidation, there is what? Organification, and then there is coupling. All right. So in this process, all of these things. So in this lumen, now all of these structures that are here now forms what we call our colloid, right? Forms our structure known as colloid. So now that is why we say that. So we have seen how these thyroid hormones are synthesized and they are stored within the luminal aspect. So how are they released? Now I wouldn't want to go into details of how these guys are released, that is whenever they are needed in the body, how they move back again to the blood capillaries. But just know that through the process of pinocytosis, right, through the process of pinocytosis, that is the luminal aspect invaginates, right, something like this, right, invaginates, and then some of this colloid, right, or some of these structures here enters into it, and then with the help of some enzymes, then some of these proteinous substances are cleaved, and then they move via exocytosis into the blood stream. So basically, that is the run through of thyroid hormone synthesis, storage, and release. Now, in my next lesson, I will be talking about the function of this T3 and T4 to end this lesson. So what is actually the function of T3 and T4? We have seen how they are produced, we have seen how they are stored, and how they are released. So for the function of T3 and T4, I will divide it basically into two parts. Okay, so this aspect I will talk about metabolism, all right, metabolism. Then here I'll talk about growth. Okay, so this is the two major functions of T3 and T4. Now for metabolism, they help in the catabolism, right? Catabolism of carbohydrates and fats. Okay? Alright, when I talk about catabolism, I mean the breakdown of what? The breakdown of carbohydrates and fats to release energy, basically. Then another function is what? Anabolism. Anabolism of protein. Right? Anabolism of protein. Then another function is that they help to maintain the basal metabolism, basal metabolic rate. So they maintain what? They maintain the basal metabolic rate. Okay? Then very, very important, they help to what? To generate heat. Alright? Since we say that they have something to do with metabolism, they help to what? To generate heat. And that's why usually if you have low concentration of these hormones, okay, call it hypothyroidism, usually those people they cannot withstand heat, so they generate what? Heat. Very important. Then they also help in oxygen utilization. Oxygen utilization. So all of these are functions of what? T3 and T4. I said what? The breakdown of carbohydrate and fats. Then anabolism, that is build up of proteins. They maintain basal metabolic rate. They help to generate heat, right? Particularly in the heart, in the skeletal muscles and some other structures. Then they help in oxygen utilization. Now, for growth, all right, for growth, this T3 and T4 act on most cells of the body. They act on what? On most cells of the body. Now, but particularly They act on skeletal muscle and bones. All right, bones. So together with growth hormone, if you remember, growth hormone that is produced by the anterior pituitary gland, they help to also regulate growth and some aspect of development. So they are also important in the maintenance of the central nervous system. Okay, so basically that is the function of T3 and T4. In our subsequent lesson, we'll be looking at disorders of these hormones. I hope you enjoyed this lesson. Thank you very much.